Hi class, today we are going to talk about the digestive system. We will talk about the organs and the general functions of the digestive system and then move on to the general histology of the GI tract and the small intestine. The main focus of our lecture today is to learn about the detailed histology of the ileum. The system of our body that helps us to digest food, absorb nutrients, and get rid of all the undigested wastes is called the digestive system. Anatomically and functionally, the digestive system is made up of a long, hollow, tubular gastrointestinal tract, also known as the GI tract, highlighted in green here, and there are some accessory digestive organs. The organs that make up the GI tract include the oral cavity, pharynx, esophagus, stomach, small intestine, and large intestine. The accessory digestive organs that appear as outgrowth of the GI tract include the teeth, tongue, salivary glands, liver, pancreas, and gallbladder. All these together help in the process of digestion. Now, after we swallow food, the esophagus transports the ball of food to the stomach, where the food gets mixed with different gastric juices. The partially digested food then moves to the small intestine, where further digestion takes place. Small intestine is also the main site for nutrient absorption. The undigested materials then move into the large intestine, where they are compacted to form fecal matter. Moving on to the general histology of the GI tract. The wall of the GI tract from the esophagus to the anus is made up of 4 layers, which can be very well seen in a cross-section. Here, I would like to show you a picture that I usually draw in my regular class. So, we are looking into the cross-section of the GI tract. We can identify the lumen at the center through which food passes, and we see the peritoneal cavity outside the tube. We will identify the layers from superficial to deep. Most superficially, we have the serosa. Deep to that, we have muscularis externa made up of smooth muscles. Deep to that, we have submucosa, which has blood vessels, nerves, and lymphatic vessels, and I like to say that the submucosa has all the stuff of the GI tract. The deepest layer is the mucosa, which has 3 components. The layer lining the lumen is called the epithelium, which is highlighted in blue. This is supported by a layer of connective tissue called lamina propria. Superficial to that, there is a thin layer of smooth muscle called muscularis mucosae. So, to remember that the layers of the GI tract from superficial to deep are serosa, muscularis externa, submucosa, and mucosa. And mucosa itself has 3 components. Don't forget. Now, remember, these layers do not look the same throughout the GI tract. There are variations to this general pattern as we move from one GI tract organ to the other. Alright, so moving on to the small intestine. Learning about the histology of the small intestine is the main focus of our discussion today. However, before diving into the histology, it is really important to talk about the general functions and the basic anatomy of the small intestine. Let's start. The small intestine is a site for digestion and major site for nutrient absorption. The small intestine is divided into 3 segments. First, we have the C-shaped duodenum, followed by the jejunum, and then we have the ileum. Let's move on to the histology now. If you make a cross-section of the small intestine, we can see the lumen in the center, and we can identify all the 4 layers from superficial to deep. We see the serosa, muscularis, submucosa, and mucosa. Now, if you take a little piece of the intestinal wall and look under the microscope, this is what you see. Here in figure B, we can identify all the 4 layers. And pay attention to the mucosa and submucosa. See how they are folded to form specific structures called circular folds. The circular folds increase the surface area for digestion and nutrient absorption. In the same figure, if you notice the mucosa, see how it's further folded to form finger-like structures? These are called villi, singular, villus. These are microscopic structures that further increase the surface area for digestion and nutrient absorption. Now, remember, the mucosa has 3 components, right? Can we see those? Of course, yes. We just need to move to a higher magnification. Let's do it. So in C, we are zooming into the mucosa where individual mucosal fold is called a villus. We can clearly identify the epithelium supported by the lamina propria, and then we have the muscularis mucosa. With this information, I want my students to remember that in the small intestine, we do see all 4 tunics, and there are specific modifications like circular folds and villi that increase the surface area for digestion and nutrient absorption. Are we good? Any questions so far? No? We're good, right? Okay. Now for lab today, we will look into a slide of the ileum, which is the last part of the small intestine. And at this point, I really want you to think and put together all the information that we have learned in lecture. Now, this is a slide that we will go over. First, let's identify the lumen. Even though this is super low magnification, we can still identify some basic structures. And in order to proceed, I will give you a little cheat on the top left corner. Looking at the cheat, can we identify this layer? Most superficial. If you think this is serosa, you are good. What is this layer? This is muscularis externa. Very good. Then this layer with all the stuff, this is submucosa for sure. And then the deepest layer closest to the lumen is called the mucosa. Highlighted in the blue boxes, we see circular folds which are made by mucosa and submucosa. And then the finger-like projections of the mucosa are called villi. Alright. Now we will zoom in and move from the luminal side to the peritoneal side, and we will zoom into each layer as we move along. We will start with the mucosa first, and we will identify the structures, and then we will talk about the specific functions of each structure. Are we ready? All right. So here we are zooming into the mucosa, and at this magnification, we are zooming into the mucosa. We are zooming into the mucosa, and recall that the mucosa is made up of 3 components or 3 layers. Looking into the cheat on the left-hand side, we can clearly identify the epithelial lining supported by the lamina propria, and we also see the muscularis mucosae. Now, we will zoom into the epithelial lining, and when we do that, we see one layer of beautiful, simple columnar epithelial cells. If you look at individual epithelial cell, you will see that the apical surface is folded to form bristle-like structures called microvilli. Now, remember one thing. We cannot see individual microvillus under a light microscope. But together, they appear as a thin border called brush border that increase the surface area for nutrient absorption. What else do we see? We also see individual goblet cells, and Now we are moving on to the lamina propria. If we look at a higher magnification, we do notice that the lamina propria is made up of areolar connective tissue. The lamina propria, the invaginations of the villi are called intestinal crypts, and the lamina propria also houses these glands called intestinal glands. Most importantly, we see these huge lymphatic nodules called Peyer's patch associated with the mucosa of the small intestine, specifically ileum. We also see the beautiful muscularis mucosa where the smooth muscle cells can be seen super clear. So moving on, so now that we have identified all these different structures, let's talk about their functions. The simple columnar epithelial cells that are found in the epithelium, they absorb nutrients and also secrete a lot of things. And also they absorb nutrients and also secrete a lot of stuff. Then the brush border increases surface area for digestion and nutrient absorption. The goblet cells produce mucin, which dissolve with water to form mucus, and that is really important for lubrication as the digested nutrients are absorbed and the undigested materials are left behind. The lamina propria is made up of areolar connective tissue, and it has blood vessels and lymphatic vessels, not seen in this picture. But understand that the nutrients that are being absorbed by the epithelial cells then are transported by these blood vessels into the larger vessels that are present in the submucosa. The intestinal glands secrete mucus, enzymes, hormones. The Peyer's patch play a major role in immune defense as they protect our body from growth of pathogenic bacteria. The muscularis mucosa helps in local contraction. I like to look at it as, let's say, a piece of potato chips is sticking to the villi. Then this muscularis mucosa will generate a contraction strong enough to flick it back into the lumen for proper digestion. Alright, moving on to the submucosa. As you can see, the submucosa has a lot of stuff. We can clearly see some dense irregular connective tissue and also some loose adipose connective tissue here. I have labeled a couple of blood vessels for you, and also we see nerves here. The submucosa is the cementing material between the mucosa and the underlying muscularis layer. Okay, the next we have the muscularis externa. Notice that it is made up of two layers of smooth muscles. Contraction of this muscularis externa allows the food to move along the GI tract and also helps the food to be mixed with different digestive juices. The two layers are inner circular layer and outer longitudinal layer, and in between the two layers there is a blanket of nerve plexus that actually control the contraction of this layer. Alright, the most superficial layer is serosa, which is made up of areolar connective tissue. It supports blood vessels, and this layer prevents friction between the organs of the GI tract as they move and function. In this layer, we can also see some collagen fiber bundles, and at the end, I want you to understand that